You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Dr. Aubrey de Grey. He's a biomedical gerontologist who's devised a platform and a research foundation called SENS, S-E-N-S, which is Strategies for Engineered Negligible Senescence. He got a BA in computer science and a PhD in biology from University of Cambridge, and he's editor-in-chief of Rejuvenation Research and a fellow of both the Gerontological Society of America, the American Aging Association. And we're going to talk about Aubrey's research. So, Aubrey, thank you for coming.
My pleasure. Thanks for having me.
Yeah, what is it about aging that fascinated you and made you want to work in the field for so long?
Work in this field was that hardly anybody else was working in it. What still completely astonishes me is that it has not always been obvious to everybody that aging is a medical problem, that is the number one problem facing humanity these days, because it causes more suffering than anything else, could be a rather high priority.
Why do you think that people haven't focused much on aging at all, even though it's essentially the number one killer of, or the number one negative health outcome for people?
Yeah, I think there's only one explanation, which is that while we're in this state where we can't yet bring aging under medical control, it is this terrible, terrible thing that's going to happen in the relatively distant future for most people. And they've got to find some way to put it out of their minds and get on with their lives. And the only way to really do that is to have some kind of irrational attitude to aging that essentially, lets you believe that it's a blessing in disguise of some kind. Of course, thinking that it's not a blessing, but that it is a complete inevitability that medicine will never be able to control, that is another way of putting it out of your mind, but it's equally irrational. It just makes no sense. And, until I was in my mid-20s, I totally assumed that nobody would possibly think that way, and that everybody would actually think it was something we ought to fix, and that biologists would be really trying to. But then I met, when I was 26, and married a biologist, and I found that wasn't true. And it wasn't just her, it was all the other biologists I was meeting. They all had this crazy idea, same as the rest of the world. So I thought, well, I'd better switch fields. Before then, I was working in artificial intelligence research, because I thought that also, there's a big problem there. The fact that we have to spend so much of our lives doing stuff that we would not do unless we were being paid for it. So we should have more automation. But of course, the problem of aging was far more serious. And so I switched fields.
So what's a sampling of some of the beliefs that you've heard from people about aging?
First, either like they are practical things, like it's inevitable, there's nothing we can do about it, everything ages, or they are philosophical, like, death gives meaning to life, or they are social. They will say, well, okay, yes, maybe we could do this, but it would create new problems that are worse than the problem we're solving. Like it would create unmanageable overpopulation, or dictators would live forever, or we wouldn't be able to pay the pension. So none of these things stand up to the faintest scrutiny, but they're still very popular.
So is your research concerned more with why we age, or how we age, and how to slow, halt, or reverse aging?
It's really focused on the last of those. So why and how we age are things that we already understand pretty well. And in fact, we already did understand them pretty well 20 years ago when I got into the field, or more than 20 years ago now. So really the goal here is the last of what you said, to use that understanding to design and develop therapies that prevent the health problems of old age. And that's what SENS Research Foundation does.
Okay, so how many heads is the aging monster? I've heard there's 7 ways we age, 9 ways we age. How many ways have you believe that are valid? And maybe if you could tick off at least some of the ways.
Right. So, well, actually, it's a lot more than that. We have a very large number of different types of molecular and cellular damage that accumulate throughout life in various tissues. But what you're referring to, these much smaller numbers, is a kind of classification of those many types of damage. And that classification is actually really important, because what it does is it helps us to identify generic approaches to repairing these various types. For each category, there's one generic repair approach. Now, different scientists have come up with different classifications, but the general principle is always that. So I was the first person to propose that any such classification could exist that could be useful. And it took me a while, actually, to really persuade my colleagues that I was right. But now, yes, as you say, different people come up with different classifications, but they're all saying more or less the same thing.
So what are, to you, there always seems to be a Pareto, even if there's many things that can happen as you age, for instance. So in your mind, in the Pareto, what's the most important or most influential effects that are going on that are causing our aging?
Oh, you really can't rank them, because, you see, evolution has developed genetic machinery, to slow down the rate of accumulation of each of these types of damage. And that machinery, the selection, the natural selection to develop and to maintain that machinery, relies on the age at which the machinery is outrun, is overwhelmed, and we get to a point where the damage actually makes us sick. Now, that means that for different types of damage, it's going to be the same age. If one of those types of machinery, of anti-aging machinery, were, if you like, unnecessarily good, then it would not be subject to selection, and it would accumulate mutations that made it work less well. So we end up with a situation in which all of these types of damage that accumulate reach a point of being pathogenic, in other words, making us sick, at more or less the same age. So we can't really say that one of them is more important than the other or anything.
Have we expanded our theory of aging to include epigenetics, the continued, let's say, methylation of and modulation of gene expression over an organism's life, and then maybe also the microbiome? It's not our somatic cells, but maybe it plays a big role in aging. Are those concepts now included?
Sure. Let me address both of those things, because the answers are not the same at all. The role of epigenetic changes in aging has been recognized since the early 1980s, but it's not quite what you're probably thinking of. What you're probably thinking of is the epigenetic clock that we see that have been very much in the news over the past several years. These changes to methylation that occur progressively in tissues throughout life. Now, it's quite likely that those things are not actually causal. They do not actually do harm. Rather, they are simple readouts of other things that are doing harm. And that's fine. That's still really useful, because if you can affect the way that aging is progressing, and those things are readouts, then you will be able to tell that your therapy is working by looking at those readouts. So that's all good. But the type of epigenetic change that I said was first discussed in the early 1980s, is what's better called epigenetic noise. In other words, it's not a coordinated change in all the cells in a tissue. Rather, it's random changes that occur through lack of fidelity of the processes that maintain the epigenetic state of a cell. And those changes certainly do accumulate. They were first discovered when people noticed that neurons in the brain in older people were expressing, albeit at a low level, genes that normally only get expressed in the blood. So, this became a thing. And certainly epigenetic noise can contribute to cancer. So it's definitely something that has been thought about and we care about. The microbiome, it's a rather different story. And my personal view is that the microbiome is something that we don't really need to pay all that much attention to in relation to aging, basically because it is very malleable. In other words, changes that occur in the microbiome during life are changes that are brought about by changes in our actual cells, in the human cells, and they respond to those changes. In other words, if we were to revert to those changes and make the human cells in the gut more useful, then the microbiome would very probably restore itself spontaneously. So we probably would not have had to do it ourselves.
I have a kind of a strange question. When do you think we begin aging? Do you think that, from the moment that, two cells unite to make a blastula that we're starting to age, or do you think when we're born we start to age, or when we get to a certain age we start to age? What does that curve look like to you?
Oh yeah, it's pretty much in embryogenesis early on. I mean, basically we can define aging as the accumulation of damage, and in particular of self-inflicted damage that is created as a side effect of being alive, in other words, of normal metabolism. And of course, normal metabolism begins right at conception. So, absolutely, damage accumulates. If we look at the reasons why people are living longer now than before, then, of course, if we look like 200 years ago, then the answer is that we are better at stopping people from dying early in life from infections. But if we look at the last 50 years, say, we've seen a significant increase in average lifespan even in the Western world. And that has, in most people's view, come about very substantially because of better prosperity leading to better prenatal nutrition. So that babies who were born, let's say, 80 years ago were simply born younger, biologically younger, less damaged than babies that were born 130 years ago.
Interesting. Okay. Do you see that continual supposed improvement in prenatal nutrition through today, or did it stop and reverse at some point? What's your opinion?
Oh, well, I mean, for sure, the obesity epidemic that we're seeing across the Western world, and especially in the US, is a bad thing. But that's what people do after they're born. I'm not so sure that it has a huge effect on prenatal nutrition, but of course it would have some. If you're a pregnant mother and you've got a poor diet and you're short of certain vitamins, that will certainly feed through into suboptimality of the metabolism of the fetus. So yes, absolutely, this is important, and it may have some contribution to the leveling off of lifespans that we're seeing right now. But really, the main source of that leveling off is simply that there is an optimum amount of quality of nutrition, and once you're nearly there, you can't do much better. Very much in the same way that we can't extend average longevity in the developed world significantly further by better vaccines or better antibiotics, because hardly anybody dies early in life from infections anymore anyway.
So do you think that we can extend lifespan or just health span?
Oh, we can definitely extend lifespan as well as health span. Of course, we will extend lifespan as a consequence of extending health span, but there's no hard limit that we can't break through. The way that we'll do it, though, is by very different types of medicine than what have been used so far. The medicines that are going to work are medicines that repair the damage I've been talking about. these various molecular and cellular changes that the body can only tolerate a certain amount of. If we're not going to be able to stop those types of change from occurring, from being created as consequences of metabolism, but we can stop them from reaching a problematic level of abundance by periodically going in and repairing most of the damage. And that is precisely the kinds of therapies that SENS Research Foundation and now a lot of other groups are striving to develop as soon as possible.
Yeah, so what are some of the efforts? What are some of the mechanisms that you're targeting? Can you be specific at all?
Sure, absolutely. Well, the best known is undoubtedly stem cell therapy. Stem cell therapy is a repair strategy. The type of damage that it repairs is basically loss of cells. So if you've got an organ and cells are dying and they're not being automatically replaced by the division of other cells, then obviously the number of cells in that organ will progressively decline, and eventually there won't be enough cells for the organ to do its job, which we can alleviate simply by injecting stem cells that know what to do, that we've prepared in the lab to be the right kind of stem cell that will divide and transform themselves into replacements for the cells that the body is not replacing on its own. So, for example, in Parkinson's disease, Parkinson's disease is driven by that type of damage, by the death and non-replacement of a particular type of neuron in a particular part of the brain. And even 25 years ago, when stem cell therapy was first tried for Parkinson's disease, it was occasionally spectacularly successful. And that was even though we had really very little understanding back then of how to manipulate stem cells so that we were injecting the right type of stem cell. Now, we've got much more understanding of how to do that, and half a dozen clinical trials are ongoing or starting up right now, and people are very optimistic that this will be a genuine cure for Parkinson's.
Where do the efficacious stem cells come from? Is it from the same person? You're just culturing more of them that exist in a given tissue than are currently there? Is it banking younger versions of your own cells and using them when you're older? Is it from donors? Is it from fats? Is it from the particular tissue? It sounds like there's many sources, but which are the best working so far?
Yeah, you're quite right. There are many sources, and they have pros and cons in terms of ease of manufacture, in terms of ease of prevention from being rejected by the recipient, all those kinds of things. But at the moment, especially over the past 10 years or so, it's become a lot easier to do this. First of all, there is the wonderful thing called induced pluripotent stem cells, a technology that was developed in the mid-2000s in Japan, basically where we can take cells from a person who needs them, we can take cells out, even if they don't have the right type of stem cell, so we can't just expand them as they are, we can take a different type of cell from that person and wind back its developmental clock so that it becomes a very versatile stem cell again, like a stem cell that only normally exists in the embryo. And then we can essentially re-guide that cell to become the right kind of stem cell that we need. And of course, it's still the same DNA in those cells that it was when we took them out of the person. So if we put them back into that same person, then they don't have a problem of being rejected by the person's immune system. Now, that's quite elaborate, though, and doing that whole process every time for every patient individually is very laborious, and a lot of people think it will never be economical. So there's also a lot of interest in allowing cells to be used that come from a different person. And then you've got two choices. Either you have the same situation as you do with organ transplantation, where you just have the person on lifelong immune suppression, or you do some clever tricks to the cells so that even though they're from the same person, they don't get seen by the immune system of the person receiving them. And one trick that actually a company I'm involved with called Agex is using for that is to mimic the tricks that happen during pregnancy, where the fetus needs to develop without being rejected by the mother's immune system. There are particular genes that the fetus makes, proteins that the fetus makes, that protect its cells from the mother, and we're trying to piggyback on that.
What about studying cancer? Cancer seems to set up a microenvironment that can, fool the immune system pretty well. maybe perhaps the lessons learned there?
Well, yeah, kind of. Yes. So cancer, of course, is part of aging. Almost all cancers are predominantly ones that affect the elderly. And so it's very much part of the area that we're looking at. And you're quite right. There's a great deal of interest in how how cancers protect themselves from the immune system. We would definitely die of cancer, probably before we were adults, if we didn't have such good immune defenses that detect and destroy almost all precancerous cells before they get to be a proper cancer. But of course, eventually, cancers get clever enough over time with enough of the right kinds of mutations so that they can evade it. And yes, you're quite right. We could, in principle, because we do understand quite a bit about how cancers do this now, and that's why we're getting so good at cancer immunotherapy, we could somehow create those same tricks in stem cells. But we kind of wouldn't want to, really, because then we wouldn't be able to use these clever cancer immunotherapy tricks that we have, or rather, if we did use them to stop the person from getting cancer, then the same therapy would also kill the stem cells that we had put into the person for a good reason.
So in addition to, effectively culturing stem cells, the ones we need, deploying them so they don't act systemically, but they act locally, any other additions to the stem cells? And I've heard of people trying to piggyback certain molecules so the stem cells will carry them to the right site and deploy them. I mean, is there any way of modifying stem cells so that they're more effective at their job?
Oh, absolutely. Yeah, I mean, you're quite right. And in fact, before I go directly to that question, let me put it in context, because actually a lot of the stem cell therapies that are done today and that seem to be somewhat effective are done with allogeneic cells, in other words, cells that come from a different person, and without any of these tricks to prevent the immune system from destroying them. So we expect that these cells will actually only be rather short-lived in the body. And one might think, well, okay, what's the point of that? The point is that before they get destroyed by the immune system, these cells actually secrete stuff that's good for their environment. And so they kind of rejuvenate the host cells, the cells of the recipient, into a manner that they can act, younger, even after the stem cells themselves have been destroyed. And of course, yes, absolutely, one could enhance that property, that beneficial property, by making various genetic modifications. Of course, the genetic modifications might be very specific to a particular tissue, to secrete such and such a molecule that maybe the tissue doesn't make enough of late in life. Absolutely.
Do we know why some tissues in the body have a lot of stem cells that are fast dividing, some have very few, and I don't know if any have none, but why is there such a difference in the amount and efficacy and rate of stem cell use and production?
Well, first of all, let me correct what you said. The stem cells in the body do not divide rapidly. In fact, that's very important. If they did divide rapidly, they would stop being stem cells. They only divide very, very slowly.
Even in skin?
Even in skin, yeah, in the skin or the blood. What happens is that the stem cells themselves only divide once every few months. But then the process of going from a stem cell to the terminal cell, the cells that actually make the skin or make the blood, that involves a lot of cell divisions during which differentiation into the eventual form happens step by step. And during those steps, the cells, which are called transit amplifying cells, those cells do divide rapidly. And that's how we can actually replenish the blood. The blood's got a lot of cells in it, and we have to turn them and they only last a few months. The red cells, for example, those cells have to be replaced at a huge rate. The way we're able to do that is by virtue of these rapid transit amplifying cell divisions. But the long-term repopulating cells, the stem cells themselves, only divide very slowly. All right, but back to your question. Why do some tissues have more stem cells than others? Absolutely. And in fact, in most tissues, like, for example, the liver, the stem cells don't divide at all, except when the liver is injured. Normally, they just sit there hanging out. They basically, at least they don't divide to a detectable rate. So they're there to respond to injury of some kind. then in some tissues we seem to have no stem cells whatsoever, or at least no active ones. So in the brain, for example, there is one region of the brain in the hippocampus where there is a small amount of neurogenesis, of stem cells dividing and differentiating to form new neurons, even in adults. But in most areas of the brain, there's absolutely none. And we'd like to fix that. We'd like to not only do this for Parkinson's disease, as I mentioned earlier, where you just have to put stem cells into one very small part of the brain called the substantia nigra. We'd also like to do it across the whole brain, because in Alzheimer's disease, for example, there is loss of neurons, across the whole cortex. It happens more in some places than others, but it's very distributed. And we've actually got a project that we're funding at Albert Einstein College of Medicine looking at ways to do that. It's a technical challenge for sure, but we think we can.
You spoke about a number of clinical trials that are in progress. Are you able to talk about what the goal of the trials are? I guess these are the closest things we're going to get right now to actual therapies that are out in the field.
Sure. I mean, the goals, of course, are to rejuvenate the organ that is being that's being affected. So in the case of Parkinson's disease, the goal is to eliminate Parkinson's symptoms. And that's certainly what seems to happen when these particular type of neurons called dopaminergic neurons are replenished. Their number is restored to useful levels.
Okay. Any other clinical trials that you think are going to be really fascinating if they're working out?
Oh, sure. I mean, let's actually talk about other types of damage as well, because these are coming to clinical trials now as well. So right now, just middle of last year, I guess about a year ago, a company named Unity Biotechnology, which is the leader in looking at what are called senescent cells, they started a clinical trial to remove senescent cells using drugs that they had developed. Now, the clinical trial is only looking at cartilage at the moment. It's treating osteoarthritis. But senescent cells occur all over the body, and we want to remove these cells because they are definitely a type of damage that's bad for us. And it's certainly the intention of Unity and other companies to remove senescent cells across the whole body. They're starting in the knee, but there's plenty more where that comes from. And a number of the other areas, including companies that we've spun out as startup companies, are planning clinical trials over the next year or two that will remove other types of damage, like molecular waste products that accumulate inside cells, for example. We've also got a company that we've spun out that is likely to be able to start a clinical trial next year on removing amyloid from various tissues, especially from the heart. Amyloid is a type of molecular waste product again, but it's one that accumulates in the spaces between cells and stops cells from communicating properly. So again, that's very important.
Maybe a couple of questions about senescent cells. What characterizes a cell as being senescent versus not?
Sure. So the main thing that characterizes it is that it is doing more harm than good by secreting into its environment chemicals that are toxic to other cells. And the second thing that characterizes it is that it is able to protect itself, very much like cancers, from the immune system. The immune system is actually pretty good at getting rid of these kind of aberrant, dysfunctional cells, but it's not 100% perfect at it. And that's because they also develop these mechanisms to protect themselves. So a lot of what's being done now is developing ways to overcome that protection. And again, that's something that we have a project looking at doing in new ways. But of course, it's also possible just to directly kill the senescent cells, to overcome the mechanisms that they are using to keep themselves alive. And that's what Unity are trying to do.
Do these cells appear in all tissues across the body, and do they appear in varying amounts? Any tissues where they seem to appear early and often?
Varying amounts for sure, but yes, all over the body, or every kind of tissue gets these things, and they accumulate steadily with age. At this point, we're not even sure whether that accumulation is because the immune system gets less good at destroying them, or whether they're just coming into existence more often because of more damage to the cells, or because they just accumulate because they're not being got rid of, and some of them are really have hung out for decades. But for whatever reason, whether it's a combination of those three, we certainly do get more of them in late life, and they're definitely bad for us.
Well, what about which tissues in which they appear would be the most deleterious, and which tissues would be the most easy, theoretically, to clean up?
Well, the reason why Unity have gone for the knee is because, for sure, there's good evidence that that senescent cells accumulate in cartilage. The cells in the cartilage that make cartilage are called chondrocytes, and actually, there's some evidence that chondrocytes are the single cell type that is the worst affected by this type of problem. The highest proportion of chondrocytes become senescent late in life. And yes, they definitely cause cartilage to be less good than it would be at regenerating. And another reason why Unity has chosen osteoporosis first is safety. Of course, it's early days, and the drugs that are being tested are experimental. That's why we need clinical trials in the first place. So if you're injecting into the knee, then it's not going into the circulation. It's got less opportunity to be toxic somewhere else, for unidentified reasons. But really, the answer to your question is that everywhere that we see senescent cells, they seem to be doing harm. The reason why Unity are doing so well, they got a huge amount of money, like $300 million invested in them a year ago, is because the results that came out of laboratory experiments done by some of the scientific founders of Unity were so impressive. You could take mice that had a lot of senescent cells, and you could genetically remove them by a trick that you couldn't actually do in humans. And they would be, all manner of ways they would be more useful. Even their spinal cord would be straighter. They wouldn't get so hunched. That's something that happens to mice normally. And their skin would be better, and their, everything would be better.
So we've been able to do that just in individual populations of cells, but not inside the body for sure, and not even in animal models.
Oh yeah, definitely we can do it. I mean, so the original experiments that were published back in 2011 were with an artificial model that was genetically modified in a way that made it easy to get rid of the senescent cells. But of course, the next step was to figure out ways to do it in genetically unmodified animals, and that's where Unity came from. Basically, they were able to identify drugs that were reasonably effective in mice, and of course that's gone on because they've been able to get so much funding. They've been able to do a large amount of drug screening and get drugs that are progressively more effective. And yes, absolutely, all over the body.
So in doing such a thing and removing the senescent cells, what was noticed? How did the creature that it was done in react?
So like I said, everything was better. You could compare the animals and at an age when the animals with the senescent cells were looking really ropey, they had bad skin, and they were not running around so much, and their spines were bent, and they started getting cancer, even things like that. None of these things were happening at that same age. The mice would last longer before going downhill.
And was this looked at, over a period of time? Did the appearance of new senescent cells happen at the same rate? Did they slow? Was there how long did this effect last? Did it just literally reset the organism back to this earlier state and it progressed forward normally?
Well, of course, there are other types of damage that are accumulating in these mice, same as there are in humans. As I said, there are many different types of damage. So, of course, the animals did get sick eventually in some other ways. But yes, these drugs that were developed were applied multiple times. So there was never an accumulation of senescent cells after that.
Are there certain types of damage that need to be addressed along with other kinds of damage in order for this to be effective? Ideally, you'd want to address every form of damage, I'm sure, at the same time. But are there a few juicy ones that you can pick that really will have a great effect, or do you need to try to affect as many as possible?
Yeah, so that's not really the way we think about it, because as you say, we do eventually have to repair everything. And so actually at SENS Research Foundation, we have put ourselves in a position where we're able to work on the most difficult areas of damage, the areas that, there's a lot of work to do, and it might even be a long time before it's possible even to publish anything. So, it's not conducive to the environment that exists in industry or indeed in academia. in terms of short-termism. And so we've been very successful in causing the most challenging areas that were initially being very neglected, causing them to catch up. We've been able to fund them and actually cause them to get to a sufficient proof of concept that we could even, in many cases, spin them out into startup companies. So yes, I don't really have a priority list, but of course, when one is looking at the development of all of this, one has got to develop the repair strategies individually, and so when one moves to clinical trials, one specifically identifies a patient group that appears to be particularly suffering from one particular type of damage, and then one can observe benefits even when one has not yet developed the other types of repair. So, the case that we were talking about earlier of Parkinson's disease is a fine example. People with Parkinson's disease have other things wrong with them as well, but perhaps not so seriously. So one can see a great improvement in cognitive function and therefore in quality of life.
I just wonder what the effect of repairing one type of damage is in the context of all these other kinds of damage. Let's say you take a 70-year-old person. They have a lot of damage in all these areas. You fix one area. How do they respond versus trying to repair 3 or 4 types of damage they have? Or let's say you take a 70-year-old guy and you give him a 20-year-old's liver. You somehow just give him the transplant and there's no autoimmunity. But what do you think would happen in that context?
So, it's just like I said, the patients that will benefit the most from individual therapies are the ones who, for whatever reason, have a particular type of damage occurring and reaching a problematic level more quickly than the other types of damage. Now, people who live unusually long, people who get to, let's say, 90 or 100, those are people who obviously do not have that problem. All of them are generating damage of all types slowly. And therefore, by definition, they're going to end up getting multiple types of damage more simultaneously than people who got sick at a younger age. And as such, they will have less opportunity to benefit from individual therapies. They will need the more comprehensive package.
Interesting. When senescence has been studied in people, have the very old been studied? Have a big range of ages been studied? Like, how extensive has our investigation been into what senescence looks like in different people at different ages and different kinds of people?
Yeah, there's not really been enough of that yet. Of course, people have been studying the elderly to look for what goes wrong with them for quite a long time. And in particular, there have been some very important studies which are called longitudinal studies, where the same people were studied starting when they were young adults and have been followed throughout their lives so as to understand better how such things actually develop chronologically. But one thing that has been missing is a thorough analysis of what really elderly people die of. And in fact, it was only 10 years ago or so that people started to do autopsies of centenarians. Just a few, of course, but still enough to get some idea of what they were dying of. Because previously people thought, well, that's not very interesting. They did well, what the hell. So there was a very interesting discovery that something I mentioned earlier, the accumulation of amyloid in the heart was actually the number one cause of death of the extreme elderly. And that was part of why we ended up funding a group in Texas, at the University of Texas, to research ways to eliminate that stuff. And that, as I mentioned earlier, was very successful, and it's now been spun out into a company.
So, in our conversation, you don't sound very surprised about a lot of things. But so I want to ask you, what does surprise you recently? What information have you learned that you're really scratching your head about?
Well, it's actually not quite like that. It's a more information. The surprises have all been good surprises, so they're more a case of throwing my hat in the air. For example, I mentioned the development of induced pluripotent stem cells. That was back in 2006, and it was absolutely out of the blue. Now, in retrospect, we should not have been too surprised that it was relatively easy to reset cells back to an embryonic state, because the early embryo has to do exactly that thing, and anything that is genetically encoded is likely to be genetically encoded by a small number of genes. But still, it was a huge breakthrough. And then, of course, there was CRISPR, this new way of editing the genome in a very site-specific way, changing particular parts of the genome. That was, again, completely out of the blue. Then, of course, there are other things that turn up that are much more specific to particular types of damage and particular ways of repairing that damage. And they get a lot more technical. For example, one of the things that we want to do is to repair damaged mitochondria. And in about 2006, again, it was discovered that there was a particular very counterintuitive trick that could be used that would help, that was going to be an essential component of the therapy that we were trying to develop. Those kinds of things happen all the time. But what doesn't seem to happen is bad news. In the entire 20 years now since I first proposed this comprehensive damage repair approach, we haven't had a single case of a new type of damage coming in, being discovered that doesn't fit into my 7-point classification. Or for that matter, we haven't come up against any new discovery that showed that the approach to repair that I had proposed back then was not going to work. So, it's all pretty good news.
How do you, I hope this is not too personal, but how do you feel about your own aging?
I'm doing pretty well, actually. I've been lucky enough to have a lot of tests of my biological age. Over the past 15 years or so, I've been tested 5 times at really high-end tests. And it's gone really well. I always come out more than a decade younger than than my chronological age, and that's pretty nice.
Okay. I just didn't know, as you worked in this field for a while, if you have any, thought, how your thoughts have changed about your own circumstance or the circumstance of people that are close to you. You feel like an impetus to make them understand what you're understanding, or do you, I don't know.
No, not really. I feel more, it's humanity in general that gets me out of bed in the morning. It's not the fact that some particular person is getting older. It's more the fact that 100,000 people are dying every day from aging. And similarly for understanding. Yeah, I mean, at the end of the day, what matters here is to get the work done. And that only takes a small number of scientists, but not all that small, therefore it definitely takes more funding, but that means persuading people that it's a valuable thing to support. And that means, of course, not only wealthy individuals or even, other philanthropists, but also people in government, which means persuading the general public to vote for it and to lobby for it. So, yeah, that's really why I do so much media and why I spend my time running around the world giving talks.
Okay. And last question. So what do you see as possible in the next 5 years versus the next 20?
I think in the next 5 years, we're going to see some incremental improvements in our ability to repair types of damage in aging. But for the reasons that we've already discussed, I think that for the average person, those improvements will not really translate into a significant extension of healthy lifespan and therefore of total lifespan. However, what we will see in 5 years is, I believe, a very big sea change in people's expectations of what's going to happen later on. At the moment, as we mentioned at the beginning of the podcast, there's a huge amount of fatalism and skepticism, and really, not very good reasons why we shouldn't do this. And that irrationality is going to go away. It's definitely going to just be replaced completely by an attitude that this really is a medical problem and a really, really urgent one, far more important than any other medical problem. I think the reason why it's going to happen soon, like in the next 5 years, as you said, is simply because it's going to be driven by the relatively modest improvements in laboratory success, in terms of, for example, getting old mice to live longer, and the impact of those advances on what other experts are willing to say to the media. At the moment, we have this huge problem that there are maybe a dozen really high-profile experts who speak to the media a lot about the biology of aging, but of those people, I am the only one who is able to tell it like it is, because everybody else relies on public money from grant applications from the government to get their work done, and that means that they are constantly petrified of saying anything that could be characterized as irresponsible, as getting people's hopes up unduly or anything like that. So they, of course, dramatically in the opposite direction. That's going to change. Eventually, the science is going to move far enough along that it's not going to be possible to maintain the view that, it might never happen. You've absolutely got to say, it's only a matter of time. So what's going to happen after that? You asked about 20 years. I think in 20 years we have more than a 50-50 chance of really getting to what I've called longevity escape velocity, where we have done enough of the damage repair portfolio to be able to essentially stay one step ahead of the problem, to repair aging as fast as the body is generating new damage.
Yeah, I mean, there's a lot of things to talk about there with the societal consequences, but I guess for this conversation, we'll keep it to the science. Well, very good. We're just about out of time. I want to respect your time. What's the best way for people to find more about what's really going on in aging, and maybe to look at some of the things that you're working on? Where should they go? Oh,
for sure. The best place to go is SENS.org, our website. S for sugar, E for elephant, N for November, S for sugar.org. We have obviously plenty of information there about what we are doing, but also lots and lots of news and other information about what other people are doing around the world in this space. And it's written for every kind of audience. There's stuff there for extreme experts, and there's also stuff there for total novices. So, you name it, it's there. And also, of course, there's a contact form. You can write to us there. You can, if you have any questions.
So, sensology.com, right?
No, just sens.org.
Oh, sens.org. I'm sorry. Sens.org. SENS.org.
Yeah.
Got it. Okay. Well, very good. Aubrey, thank you for coming on the podcast. I really appreciate it.
Well, again, thank you for having me.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.